I think you don't know what it's like to work in a lab until you do it. And so I think that it's hard to know, do you want to go to graduate school, do you want to go to medical school, do you want to work in a biotech company? Even if you're really interested in biology, it's hard to know. Because not only is it the process of doing experiments, but it's the lab culture. And so I think it helps you figure out not just what you want to do, but what you don't want to do. The students in my lab actually get involved in experiments. So they're basically kind of two things that most students do in most labs. One is doing a lot of lab chores, washing dishes and doing autoclaving and things like that. And while that seems kind of boring and tedious, it is still a way to learn about what happens in a lab, because you meet the people in the lab, you hear about what they're doing, so that's still useful. And often what many labs will do is have students do that first and then move into a project in the lab. And so in my lab, when students work on a project, it's usually not growing the stem cells or doing a lot of the hands-on with the stem cells, but it's a lot of the analysis. So a lot of doing PCR or immunostaining or things like that, where it doesn't require so much specialized training, but it enables you to learn how the analysis is done. I mean, the outcome of the experiment. So that's usually what students in my lab do. So my lab uses human stem cells as a way to model and study developmental disorders, including Down syndrome and Fragile X syndrome. So I think that development is really cool. I remember taking a developmental biology class, and the thought that you can start with 2 cells and end up with a whole organism I found really exciting. Like, how does development happen? And so I've been interested for a long time in developmental biology, and stem cells are just the perfect way to me to study developmental biology, because you can start at the beginning and kind of see what happens. So that's why I like stem cells.
Surrounding it.
I would say I think it gets a lot more attention than is warranted, because actually now with induced pluripotent stem cells, there aren't as many researchers that are jumping into working with human embryonic stem cells. But also I think there's a big misconception that every time we go to do an experiment on human embryonic stem cells, we have to go back and make more and go to the embryo. And we all use human embryonic stem cells that have been around for 15 years. However, I totally understand the ethical considerations that come with that, but I also think it's very regulated. So one is it avoids the ethical debate of human embryonic stem cells, but I think that is a little bit of a minor consideration. I think what's more important is that you can, first of all, get a lot more stem cells. You can get them from many different people, and so instead of having a pool of, say, 20 human embryonic stem cell lines, so basically those would have been 20 people, instead you can make hundreds of iPS cells from hundreds of people, and that makes more sense when you're studying human diseases, because there's so much genetic variation between people. So I think that sheer number of iPS cells that you can make makes the studies much more powerful. But more importantly, I think the fact that you can make iPS cells that carry diseases is really the most important thing. You can, whether it's a genetic disease or not, so something like autism, you may not know the genetic basis, but you can get cells from an individual who has autism. You know what their characteristics are, and you can see if you can find something in those cells in the culture dish that might give you some hints as to the cause or the consequences of those disorders. So I think that although most people think about the therapeutic value of stem cells primarily in transplantation, I think those kinds of studies, that might be a little bit farther off, except for maybe some very specific diseases, like maybe those of the eye, where it's really accessible and easy to transplant. But I think that what we'll see much more of in the next 10 years is using stem cells for disease modeling and learning about different diseases. I think that's really moving very fast. And so, for example, in the kinds of things we study here at the Waisman Center, neurodegenerative diseases and neurodevelopmental diseases, there aren't really good ways to study, or stem cells provide a really great way to study these things. And I think we can learn a lot about the causes of these kinds of disorders with the stem cells. So I think that